All right, listen up, students. Welcome to this definitive explainer on the cell cycle, mitosis, and meiosis. I'm speaking to you right now as your senior AKTU faculty member and, quite frankly, your strictest invigilator. My goal today is absolute perfection. I'm here to guarantee, and I mean guarantee, that you will not lose a single solitary mark on your upcoming unit exam. We are going to master this topic so completely that when you finally sit down in that exam hall, the answers are practically going to write themselves. Let's get to work.
Now, you might be wondering, before we get into all the heavy textbook theory, let's just step back and dive into a biological miracle that we frankly take for granted every single day. How exactly does a tiny child become a full-grown adult? Or think about this. When you get a deep cut on your skin, how does that tissue heal so flawlessly that eventually you literally can't even see a scar? How does a single cell know how to make an exact, perfect copy of itself? Well, the answer to all of these questions lies within the highly coordinated actions of roughly 37 trillion cells, all working in absolute harmony inside your body. It's mind-blowing when you really think about it.
So, here's our master syllabus for today's session. 1. The blueprint of life. 2. Phases of the cell cycle. 3. Mitosis, standard cell division. 4. Meiosis, reproductive cell division. And finally, 5. Mitosis versus meiosis compared. Let's jump right in. Section 1. The blueprint of life. Understanding the fundamental unit.
Okay, so a flawless exam answer always starts with a rock-solid foundation of the basics. Think of your body like a massive, incredibly complex skyscraper. Just as a building is constructed from millions of individual bricks, your body is built entirely from cells. The cell is the absolute smallest living unit of your body. And right in the center of almost every single one of those cells is a nucleus. I really want you to think of the nucleus as the brain, or like the master control room, because inside this control room sits your DNA, this incredibly elegant double helix structure that serves as your ultimate instruction manual. Literally every trait you possess, the exact color of your eyes, the shade of your hair, your height, your skin tone, and exactly how your body functions day to day is securely stored right there in the DNA.
Now, why do these tiny units actually need to divide in the first place? Well, there are 3 strict biological mandates for cell division: growth, repair, and replacement. When we're young, our cells must divide rapidly for us to grow taller and stronger. When we sustain an injury, like a scratch or a wound on your arm, brand new cells absolutely must divide to repair that damaged tissue. And finally, replacement. Millions of your skin cells are dying and falling off right now as you listen to my voice, and they have to be constantly replaced by newly divided cells just to keep your body protected. Section 2. Phases of the cell cycle. The biological clock. I want you to picture this as the ultimate journey of a cell from the very moment it's born until the exact moment it divides into 2. This cycle is a master process flow, an exact chronological progression that you absolutely must memorize in this precise order: G1 phase, S phase, G2 phase, and finally the M phase. If you write the sequence out perfectly in your AKTU exam, the examiner immediately knows, without a shadow of a doubt, that you understand the exact timeline of cellular life. Let's break them down step by step. We begin with the G1 phase, or gap 1. I actually like to call this the working phase or the growth phase. The cell has just been born, and honestly, it needs to get straight to work. It actively increases its physical size, it aggressively synthesizes brand new proteins, it builds vital organelles like mitochondria, and it hoards energy. The cell is essentially bulking up, preparing itself for the massive undertaking of division that lies just ahead. Now, hey, listen to me very carefully here, students. This is a classic trick question that us invigilators absolutely love to throw on the exam. Not all cells proceed from G1 into division. Some exit into what is known as the G0 phase. Now, it's called a resting phase, but mark my words and write this down: the cell is not sleeping. Do not write that it is dormant. The cell is fully biologically active. It's doing its daily job. It is just not actively dividing. Brain cells and certain nerve cells are super famous for entering G0. Remember this key distinction, and I promise you, you will secure those critical marks. All right, so that aggressive preparation builds perfectly into the S phase, which stands for synthesis. This is arguably the absolute most critical step of the whole cycle. Here, DNA replication occurs. The cell takes its single precious copy of genetic information and perfectly duplicates it into 2 identical sets. I want you to literally underline the word replication in your notes right now. The upcoming new daughter cells absolutely require a complete, identical set of genetic material to function properly.
Without the S phase, cellular division would be an absolute catastrophic failure. Right after replication, we enter the G2 phase, or GAP2. Think of this as the ultimate biological quality control checkpoint. The cell acts as its own strict invigilator here. It scans the newly copied DNA. Did it copy correctly? Are there any sneaky errors or mutations? If an error is found, the cell deploys repair mechanisms to fix it immediately. Only when the DNA is absolutely 100% perfect is the cell officially authorized to proceed to the actual physical division phase. Section 3, mitosis, standard cell division, the M phase. So the preparation is over. The blueprint is fully copied. Now the actual structural splitting of the parent cell finally begins. So the crucial point here is how you remember these complex steps. I want you to aggressively anchor this 4-letter acronym into your minds right now. PMAT. PMAT. Whenever you see a question on mitosis, write PMAT in the margin of your exam booklet immediately. Seriously, do it. Let me animate this process for you so you can see it perfectly in your mind's eye. Okay, step 1, prophase. The loose DNA tightly condenses, rolling up into thick, highly visible chromosomes. The nuclear membrane breaks down and just disappears, and microscopic spindle fibers begin to form, kind of like a spider's web. Step 2, metaphase. This is just beautiful in its precision. Every single chromosome lines up perfectly straight down the absolute center of the cell along what we call the metaphase plate. Step 3, anaphase. Action time. The spindle fibers violently pull the sister chromatids apart, dragging them to exact opposite poles of the cell. And finally, step 4, telophase. The chromosomes arrive at those poles, they begin to unwind back into thin threads, and a brand new nuclear membrane forms around each of the 2 new sets of DNA. PMEIT, perfectly executed. But wait, hold on, the job isn't quite finished yet. Actually, scratch that. We're so close, but telophase only gives us 2 nuclei inside one giant cell body. Enter cytokinesis. The outer wall of the cell, the cytoplasm, pinches inward right down the middle, exactly like a belt being pulled super tight, until it completely snaps and divides. Now, finally, the whole shebang is done and you have 2 distinct, fully independent, and genetically identical daughter cells. 46. I'm actually pausing here for dramatic effect, because you absolutely must hammer this exact number into your memory. In humans, standard mitosis is all about maintaining the exact same number of chromosomes. The parent cell had 46, and both brand new identical daughter cells have exactly 46. No more, no less. It's a perfect match. Section 4, meiosis, reproductive cell division, having the blueprint. So everything we literally just talked about with mitosis applies to your skin, your bones, your stomach. But meiosis? That's strictly and exclusively for reproductive cell division, creating the sperm and the egg. Notice the stark contrast here, 23. Now, why must meiosis cut the genetic blueprint precisely in half? Well, think about the mathematics for a second. If a sperm and an egg both had 46 chromosomes, their combination would create a child with 92. The next generation would have 184. Our chromosomes would catastrophically double every single generation. Biology is way smarter than that. Meiosis strictly halves the blueprint down to 23, so that when the 2 cells combine, they restore the perfect human number of 46. To pull this off, meiosis requires a double division. Let's trace the steps. You start with one parent cell. It undergoes meiosis 1, splitting into 2 cells. But it doesn't stop there. Oh no, it immediately undergoes meiosis 2, splitting those 2 cells again. The absolutely critical takeaway for your exam here is the final yield. Meiosis produces 4 highly specialized daughter cells, not 2. Section 5, mitosis versus meiosis compared, the ultimate cheat sheet. All right, students, consider this next bit the ultimate cheat sheet that you absolutely must perfectly replicate in your exam booklets. Let's break down the side-by-side logic. Divisions, mitosis has 1, meiosis has 2. Cells produced, mitosis gives us 2, meiosis gives us 4. Chromosome count, mitosis keeps it exactly the same at 46, while meiosis deliberately cuts it right in half to 23. And finally, the purpose. Mitosis is the workhorse for body growth and wound repair, while meiosis is the highly specialized architect of reproduction. Listen to me, if you draw this exact table on your exam paper, the invigilator will have literally no choice but to award you every single mark available. It's a game changer. But, Beyond just securing those perfect exam marks, I want you to consider the beautiful reality hidden behind all of these facts. Mitosis makes identical clones. It has to. You want your healing skin cells to be exactly the same as the old ones. But meiosis creates genetic variation. Because the chromosomes shuffle in half, every single reproductive cell is completely unique. This is the brilliant biological reason why you look similar to your parents, but you're not an exact clone of them. It is literally the engine of human uniqueness. So I'm going to leave you with this final, thought-provoking question. Without this precise, microscopic machinery perfectly executing the biological clock, flawlessly running PMAT to heal your cuts and grow your bones, would life as we know it even exist? Every healed wound, every single inch you've grown, and every new generation on this planet is entirely thanks to this magnificent process of cell division. You are now 100% prepared, empowered, and ready to totally dominate your AQ2 exam. Keep this structure in your head, remember your PMAT, and you will be absolutely unstoppable. Good luck out there.